thing, and I tell them, memorize it. And they go, huh? And I go, I'm the teacher, you're the student, memorize it. And you know, the look on their faces, this utter shock, because I've usually taken it out of some fine print brochure of some product or something. Yeah. Yeah, when I went and made the announcement, a bunch of coffee cups rustled. So.
Okay, we are on live.
I will be short. It will not take 2 hours. I mean, I will talk like 45 minutes. So we don't worry about pushing the deadline. But if they miss this, they miss the whole like aha value of the thing. Need more?
Come and get them to you, don't worry. But let's get started since it's already 11 o'clock. Okay, good morning, Happy New Year, and welcome to year 12 of Chai and Why. I have been traveling like crazy. I have not been able to put up the next few events, but don't worry, Chai and Why is happening. I don't know who went and posted that, oh, is Chai and Why happening? I mean, yes, Chai and Why will happen. So we are there every 2 weeks. I see a lot of familiar faces, I see lots of new faces. So if you're coming here for the first time, Chai and Why happens on the first Sunday of every month here at Prithvi Theatre, on the third Sunday of every month at Rewiring College in Matunga, and for months which have a fifth Sunday, we are at Alexandra School in Ford. So these are our standard venues. And increasingly, as far as possible, we will try to be online. You might want to pick up a piece of paper. And a request before we start, kindly make sure that your phones are on silent. Feel free to take pictures or whatever. Remember, if you have family, friends who are not here, you can tell them that we are live streaming this talk on our Facebook page, so you can watch it live at home. Of course, you can't participate in all the interactive activities that we do, so it's always good to come here and have more fun. Okay, so just to inform you that the next Chai and Why, 2 weeks from now, at Rewiring College, is going to be, is it the year 2020? Okay, simple question. We're going to look at calendars and how we count and how different civilizations count and how we keep track of time and years and things like this. So it's a talk on mathematics, astronomy, culture, everything put together on calendars. So that's what's coming up 2 weeks from now at Rewiring, and in a few days we'll be putting up what's happening here a month from now in February. Okay, so with that introduction, and just remember, you can follow us on Twitter at Chai and Why, Facebook at Chai and Why. So please follow us, and you can ask questions online, no problem. We will do that. Okay, so with that, it's time to get started with today's session, and we are delighted, very, very happy to welcome Professor Shubha Tole back to Chai and Why. So in this journey of outreach, come Shubha, come, so that you're on camera. In this journey of outreach in Chai and Why, Shubha
I have to do it for the camera.
Yeah. Shubha has sort of been a co-conspirator in many of the things we've done, has supported several of our activities, has come up with lots of new and interesting ideas. In fact, the last time she was at Chai and Wai, we played the ribbon game over here. We had the audience all tied up with ribbons, and I have no idea what she's going to do today. But let me introduce her. She is one of the foremost neuroscientists in India, in the world perhaps. And she is a Mumbaikar. She started off in St. Xavier's College, BSc, and then went and did a MS and PhD at Caltech, a postdoc in Chicago, and then came back about 21 years ago and set up one of the finest labs in neuroscience at TIFR, where she works on all kinds of fancy stuff that happens inside the brain. Hopefully not rat brain, mostly.
Well, if anybody will give me their brain, I will be very happy.
Yeah, so, okay, and thanks to all this Bheja Fry that she does, she's won every award there is, Bhatnagar, Infosys, etc., etc. But the main thing is she makes science fun, okay? And she comes up with all kinds of cool stuff. So I have no idea what's in store, but sit back, enjoy, and be prepared to get confused.
Thank you, Arnab. Very good friend, collaborator, and I like the word co-conspirator. And Fraser Kulkarni, who's been our partner in crime. It is so fun to bring what we love to people who may not get a taste of it, but be sure you are funding it. Your taxes fund our research. We are very grateful. We hope our research will, contribute to making everybody's lives better. But for the most part, you're paying us to do something we love. And we are deeply, deeply grateful. This is the reason why I encourage all of my students, who are also paid by taxpayer funds, finally, to do outreach. I would like for them to go to their schools, to their colleges, and to give a little taste of what they do in the lab. So here's our effort today. Anybody doesn't have a paper, you have to take detailed notes. Oh, I do this little experiment. So, okay.
Only one thing. After the experiment, please either return the papers on the table here or fold them, put them in your pocket, take them home, but don't leave them wherever they are. I mean, we cannot go around cleaning the theatre after it, and we'd like Prithvi to have us back.
In my last Chai and Wai, I threw soft balls into the audience to test reflexes. When you work on the brain, you can do pretty much anything and, call it a brain experiment. Luckily, I had student volunteers with me, and then I asked the audience to test the students' reflexes back. So that's how we got all the balls back, but please do not test my reflexes. I'm old and aging, and the brain does age. Okay, so what is this about? You may have done this experiment before, but no matter how many times I do it, it continues to fascinate me. So let's have some fun. Roll up the paper into a little light roll. Don't strangle it. Yeah, you want it to be about the size of a 1 rupee coin, and you want to hold it lightly. Old coin, new coin, doesn't matter. Eventually you have to look through it, okay? But so hold it lightly, because before you begin to look through it, you're going to practice what your other hand does. So doesn't matter which hand you hold it, okay? Just try and mirror me, okay? So keep it in one hand. So how many of you are holding it in your right and how many in your left? This is going to be difficult. It doesn't matter at all, but you will get confused because I'm going to try and mirror what you do. Weirdly, this is something to do with how the brain functions, that we can mimic somebody very easily when you mirror them, okay? If I use my right, you're going to use your left, okay? See, isn't it much easier to do copy me? And you can do this actually with small children. If you catch the eye of, say, a 2-year-old, that's the perfect age. They might be sitting across the room, but if they and you have a connection, you can actually get the child to do all kinds of funky things in mirror image, okay, if the child is interested in what you're doing. So if you see scientists sitting in, perfectly normal places like a cafeteria, doing like really weird movements, be sure there's a child somewhere that they're trying to, do the mirror experiment with. They're not just mad. Okay, so hold this. Everybody, can you hold this in the mirror image hand, which would be your right hand? Okay. Your left hand is going to be in one of two positions. Pretend this is a cylinder that's filling with water. So keep your hand like at the low level where the water is now, and then slide it along so the cylinder is full. Okay, your palm has to touch as you slide all the way up. Now, full cylinder is position 1 and empty cylinder is position 2. Okay, don't block the cylinder, you're just keeping it on the side like you're measuring its level. Come back to position 1. Okay, now just keep both eyes open and raise it so that one eye is looking through the cylinder and the other eye has just got your palm in front of it. You don't need to do the squinty thing, okay? It's okay if your lashes touch your palm. You just need to look through it, and one eye has the palm, so you know what you're going to do, that eye can't see. But can you see me through the other eye? Yes. Can you see me? So if I do the James Bond, okay, now simply slide your hand to position 2. What do you see? What do you see? What did they serve in the cafe out there? I'm hearing there is a hole in the hand. Okay, okay, is the hole maybe only happening because your palm is there? Maybe you slide up your hand like so. You can make, oh, I wish I could take a video of you guys doing these weird things. You can make the whole travel, huh? But look at it, admire it. What you see through the hole is crystal clear. It's your view of the world, right? Look at the edges, sharp. And then there's your palm. Oh my God. So what I open my graduate level neuroscience class is a debate. Okay? I open with a debate. Is the hole in the hand real or is it imaginary? Is it real or is, so let's do a poll. Am I audible and am I supposed to be on mic?
The mic is only for the webcast. This auditorium has no...
Ah, okay. So, how many people think the hole in the hand is real and how many think imaginary? Let's go real. How many? How many people, okay, how many people did not see the hole in the hand?
I couldn't figure it out.
You guys are the normal ones, it's okay. Most people. Okay, you want to try again?
I did, finally she told me.
Okay. So how many think, okay, nothing bad will happen, okay? There are no wrong answers here. You know, when you're teaching students, you have to tell them nothing bad will happen because we are so programmed to try and give the right answer, right? But this is to stimulate discussion on what it is the brain does. So how many are going for real? 1, 2, 3, 4, 5, 6, 7, 8. How many are going for imaginary? Many, many, many. How many still don't dare to guess? Okay, now if I opened this debate here and now, we would take the entire chai and why, but maybe we can do this at the end of the session. Okay, so keep this in your mind. Whatever it is certainly brought to you through your eyes and the circuitry from the eye to the brain, okay? Right, we didn't give you some extra special, you know, sort of ESP driven sort of images, right? Whatever it is you saw with your own eyes. This session is to begin to stimulate, how do you say it, self-doubt. Self-doubt. To be a neuroscientist is an exercise in the path towards utter humility, okay? You realize how much we are led by the nose, okay? We are led by our all 5 senses into thinking the world out there is the way it is. How do you know what's out there? How do you know what's out there? If you were a brain in a bowl and you were planted in the middle of the room and that brain was asked, hey, you know, do you want to know what room you're in? The brain would say, yeah, get me a pair of eyes, get me a pair of ears. Fair enough, right? You know what the world is out there through your 5 senses. And the brain is very, very good at constructing an experience for us of being in this world. The brain is so good at it that even when the information is really, really pathetically poor, the brain will still give you a sense that all is okay. I'll give you an example. You know we have a blind spot in the retina? How many have never heard the word blind spot? It's okay. Okay, where the optic nerve goes through the retina, okay, there's a whole cable going through, right? It essentially is a place where you can't have any sensors. So if you don't have any sensors, you're blind there. And there are all kinds of exercises. So look on the web and do a blind spot exercise and you will see that there is a part of the world which you cannot see. So okay, if you're looking here, the blind spot is a little to one side and you have to do it with only one eye at a time because the other eye's blind spot is somewhere else. So usually you can cover it up. So if you look in one eye, you'll see a blind spot. But what the eye does is essentially it, scans the whole area and continuously updates to give you a continuous picture of the world. There are cool experiments where if a person is set up in an experimental situation where their eye movements can be tracked and wherever their blind spot is, that element of the view changes. You can really keep changing things and the person does not know because our brain is good at allowing us to believe that we know everything that's out there. Essentially what the brain is doing is, all is well, all is well. Even when you have no idea what's out there, all is well, okay? So, you're all laughing, you're, yeah, okay, this is a scientist saying all of this, so let's give her a little bit of a chance, I just showed you hole in the hand for God's sake, okay? And many of you said imaginary, all right. I can imagine you said imaginary because there is no hole, no? How many of you are essentially thinking that? Good enough, you did a scientific experiment. You made one observation based on one set of sensors and you said let's cross check it with another set of sensors. You did good, but that's the best we can do, okay? What if your second set of sensors is also limited? I'll give you an example. I want you to, you've played tennis or seen a tennis net. Imagine a tennis net, close your eyes and use this wonderful sensor that you just used to tell me, is there a hole in this material, tennis net? Can you tell me? Is this net holy? How many go, yes, this net is holy? You're blindfold, remember. Okay, all right, your sensor works. Now I'm going to give you a mosquito net. Close your eyes and with the same sensor tell me, is this material holy? Mosquito net. How many of you can feel the holes in a mosquito net with your finger? No, you cannot use your eyes. You're blindfold. Okay, you have to do the experiment properly. You're blindfold and I'm giving you a mosquito net.
I can feel mine because mine has, my mosquito nets have, there are 3 layers, the outer ones have big holes.
Ah, he's got a mosquito filtration system. Big holes. Okay, think about the finest layer, the finest holy layer. Can you tell whether it has holes with your fingers? At the most, you'll feel it a bit rough, right? Now, instead, I will give you a toothpick. I will change your big fat fingers, fat relative to the mosquito net, okay? Please don't feel hurt anybody. And I will now give you toothpick-sized fingers. And I will ask you, is there a hole in the mosquito net? Yes. So what did you do? You changed your sensor, right? Imagine if we were to probe the world with sensors that allow us more abilities than our 5 senses. Imagine what the world would look like, okay? See, if you put an ant on a watermelon, it's going to say, my world is green and flat, because for the extent that the ant can see, it's a flat world. If you put humans on an earth, they're going to say, to the extent I can see, the world is flat. It's a question of whether your sensors are of the scale of what you're looking at, right? So, we humans, we proud, arrogant humans, have constructed our world of sizes and shapes that we can see, we can feel, and we live in, and we think that's the way the world is. But you've seen these lovely movies, animated insect movies, ants and Bugs Life and so on, where these little insects are going through this great big tropical forest and all it is a bunch of grass on the ground. That's another illustration of scale, right? Depending on the scale you're looking at, your world can be completely different. Okay, the carpet on these stairs can be incredibly difficult to navigate through if you were very small. Now, we made the carpet so, you know, we wouldn't have trouble navigating on it, right? Somebody tell that to people who put thick pile carpet in the airport terminal T2. Impossible to, I mean, you know, that pile is like the size of the wheels of the trolley, which is why you can't navigate through it, right? So we created the world the way we like it. And we're happy to think this is the way the world is, okay? I'm going to push this point a little bit to allow us to question, do we really, really know what's out there? And is there a way of knowing it, okay? One of the things we put in our world is beautiful flowers. Pretty flowers. How many people like flowers? I can't grow them, but I certainly like them. And, you know, you wouldn't argue perhaps with me with, you know, okay, there's a purple and white flower and a yellow flower and a purple flower, right? All right, were the flowers beautifully colored so that you and I could decorate our homes, give them to our girlfriends and boyfriends? Were the flowers colored for that? Evolution gave the flowers colors or properties so that they could attract insects, which would pollinate them and make more little flowers. Evolution selects for what is what can be easily propagated, right? If you can't propagate that characteristic, it's dead. Yes, sir.
I think that we use some plants are actually DNA edited by us, so they are made for us.
Ah, very clever. Oh my goodness, we have GMO, DNA edited flowers that are made for us. Perfectly fine. I withdraw my statement. All flowers were not made for insects. Some flowers were made by us. No, he's right, he's right, he's right, he's right. Okay, it's more than flowers. Dogs have been bred for interaction with humans, right? So they are this completely weird, okay. Even when you make a flower that you think is for you and it's, say, yellow, okay, it's like we're making a movie. Things are not, things on the screen don't actually happen, right? How many people think things on the screen actually happen? Okay, how many of you think a movie actually has moving things in it? A movie is a whole bunch of stills, right, like a flip book, which are shown so fast that your eyes think it's moving. So if you think a movie has actual things moving in it, see me outside. I'm a neuroscientist. Okay. My point is, we see these flowers the way they are with our sensors, our great big eyes that see the world and tell us what's out there. And these sensors are so pathetically limited that we should actually sit and weep. We could all take out our hankies and sob as to how much we don't see. Because our eyes have sensors that see only in a tiny, tiny range of the visual spectrum. Just a few hundred nanometers, that's our visual range. There's a whole bunch of frequencies out there. So, for example, the Tata Institute has a radio telescope. It's got great big 100-foot antennae, okay, some 30 of them looking up into the universe. Because there's radio waves out there, great big wavelengths that we can't see. They would pass us by, okay. There is tiny, tiny, tiny wavelengths that we can't see, okay. There is microscopy at pushing the limits of what is even detectable. Forget the limits. There is ultraviolet, which is just a little bit shifted beyond the very, very blue-violet range of what we can see. We can't see ultraviolet. We can't see a lot of what's out there. Now, I've taken these images from somebody who photographs flowers in ultraviolet light. Now, since we can't see in ultraviolet light, how is he going to show us, right? So, these images are false colored to allow us to see, but basically there are elements in the flowers that send back light with different patterns. And this is how a flower might look if you could see ultraviolet light. Good grief, right? One thing that immediately pops out is that there is a bullseye-like pattern, a center and a surround. Here again, a bullseye-like pattern. Now, bees can see ultraviolet. Okay, who was the flower made for? Most flowers. Right? if a bee were to look at a flower, it's seeing, I cannot tell you that it's seeing these colors, but it's seeing differences in the center and the surround, and it's guiding the bee in to where the nectar is, and then it pollinates and so on. In fact, this one is a little landing strip. Can you see? A lovely landing strip. So we didn't know this was there. We didn't care. We thought we knew the way the world is because, you know, we're so confident and so full of our abilities, and all is well. What we see is the only thing that's out there. Yeah. Let's carry this a little further. You may have caught this image of multicolored balls. Okay. Now, when I saw this, I was like, oh wow, somebody has gathered balls of at least 3 different colors. Okay, there's the greeny ones, there's the reddy ones, and there's the bluey ones, right? Turns out that this too is, it's an optical illusion, and it's not there to trick us. It's there to tell us that sometimes our eyes, the circuitry of our eyes is ascribing colors to things when they don't actually have that color. So these illusions are not to sort of fool you, but just to tell you that you are bound by your circuitry. Okay, this whole talk is about the circuitry of sensation, and I'm going to bring out the message that we are controlled by our circuitry. Okay, so here I've just cut pasted the 3 colors of the 3 walls right near the edge. Okay, green, blue, and browny red, and then I've just magnified it, and then I've just blocked out the colored bars running through it. Okay, I haven't changed any color. I've just edited, and you can see that the balls are actually the same color. It's the exact same ball. But when you have different colored stripes going through it, your eye transfers the color and gives you a false colored image. Okay, our eyes do this all the time in an effort to tell us the world is understandable and make sense of it. Okay, for example, when I'm standing here in front of you, okay, not you, young man, because you can see the rest of me, but most of you are only seeing half of me, right? And now suddenly there is more of me, not for you guys. So have you ever played this hoo-ha game with kids? Why do kids get so surprised? Because, okay, they have to learn to complete the image. Okay, when something is not there in a little infant's world, it just vanishes from existence. And then suddenly it comes back into existence. This has to be learned, right? That when there is something occluding an image, your brain kind of says, okay, but you know, using what it has learned, it puts together the image from when you can see the rest of me and basically says, okay, this is an object that's hidden. Babies can do this. You can really test what a baby mind is figuring out, and you can test what skills are learned and what skills the child is born with, or what skills the child knows at an early age. I'll give you an example. How could you possibly test this on babies, right? Do you put electrodes in the baby's brain? No. Behavioral psychologists have come up with a wonderful way to ask what an infant can discriminate. For example, baby math. How many people here are kind of terrified of math and like, glad we left it kind of thing? It's okay, you know, it's your brains are wired the way they are, and there are things that you guys, you know, found very easy that the rest of us found incredibly difficult. Okay, by the end of this talk, I will talk about differences in circuitry. But babies can do basic math. How would you even test this? Okay, the way you do this is you have the baby sitting in with their mom or their dad. They're very comfortable and they're sucking something, you know, they like to, and when a baby is bored, it is, and when a baby becomes interested in something, immediately, okay, that instinct is faster. So if you put a little sensor in the tube, you can tell when a baby suddenly becomes interested. Okay, if I showed you a dog barking away and going and biting, you know, somebody's leg, that's kind of all right, but you know, I mean, not all right, but you kind of know this is to happen. If instead the person it was biting turned around and started biting the dog, you go, okay, so your surprise is an indication of what you thought was expected. Yeah, so this is how you can test babies. You have a screen, and behind the screen would roll a ball, and the baby is going, ball roll behind screen. Okay? It's now past the age where an object just disappears out of existence. It kind of knows balls behind screen. Then another ball rolls behind. Then the screen lifts up, and there are 3 balls there. Baby's going, what happened? Right? So that's the way you can test whether the kid was counting. Because if it shows a reaction that expresses surprise, it's then an indication that it knew what was expected. So you can test these skills in babies, and this is what neuroscientists, neuropsychologists do to ask how does the brain achieve its abilities. Because I hope you're picking up the theme here that everything we think we get from the world isn't just something is there and now it comes into our brains. It's a highly processed signal. Here you're processing color, right? When you did hole in the hand, the nerves, the images from the 2 eyes were being overlapped in the brain. And that's how you process that signal. Now, usually the 2 eyes are looking at the same thing, okay, but not exactly in the same way. The 2 eyes look with some slight difference of angle, and the difference in these 2 images allows the brain to compute depth. This ability of the brain is exploited in 3D movies. Okay, have you all seen 3D movies without the glasses? There's a double image, right? And the glasses simply separate out the 2 images. What we did with the cylinder and the hand, 2 separate channels, is what the glasses do, red and green. And those 2 images come in through, through separate channels, and the brain overlaps them. Now, if something is very near me, the discrepancy in the 2 images is going to be very high. If something is very, very far, the discrepancy in the 2 images is going to be less. These discrepancies are coded into the image in the film, and that's how you see foreground and background. Okay? So the circuitry of your brain is nicely being fooled and challenged so that we can see 3D movies. And all of us go to these movies, 3D movie, we pay lots of money to go sit in a hall and watch this 3-dimensionality, which is just encoded differences of discrepancy, right, for the 2 eyes. In fact, we go and see a movie where things move and people laugh and play and kill each other and whatnot, when things aren't even moving, it's just a whole bunch of stills. We voluntarily go and pay our money to be completely deluded. Is this an intelligent species? Hey, what if our whole life is a movie theater that we can't leave? What if we're actually living in a matrix? Would you have any way of testing, right? So I can guarantee you will be entertained in this talk, but I can't guarantee you will be comfortable, okay? I can't guarantee that you won't leave this talk saying, what's it all for? What's it all about? Okay? And here's my punchline. If our whole life is basically what we are desperately making up from our senses, okay, and somehow trying to live with, our brain telling us all is well, if that's all we've got, hey, let's make the most of it. Okay, let's give ourselves one hell of an experience. Let's do as much as we can, okay, with our limited abilities, because if it's all we've got, then it's all we've got. Why waste it? Okay, it puts a whole different perspective on all the little petty, things that overtake our lives sometimes, right? All the little squabbles and all the things we stress about. I mean, hey, if the whole thing is a movie, is that the movie you want to watch? Okay, so I've done vision, okay, context being everything in this case. Context is everything actually applies to much more than just vision. Okay, let's do sound. Let's do some hearing. Everyone's ears all opened? This is called the Mcgurk Effect. No matter how many times I see this or hear this, I'm completely amazed at my own ears. This is high-tech challenging sound. Which none of you can hear. Sound, please.
Wait.
Yeah, this got an auto off. After a certain time, it just switches off.
Okay.
Battery 80%.
Battery 80%. Say again? Okay, one more time.
Hey, hide it.
Now it's off sync. Oh God. Okay, high tech sound, we're going to go once more. What did you hear? They all passed the test. Okay, one more. If you didn't hear ba ba ba, see me outside. What did you hear? Now, here's a test. Promise me you'll keep your eyes closed. God promise. And I'm going to play this again. Okay? You can open and look in the middle. Yup. And oh, happened. Okay, it's the same soundtrack. It's the same soundtrack. And to prove it, we're going to do this again with both images and soundtrack. You can look at the one face, you can look at the other face, you can close your eyes and test your own circuitry. That circuitry which we are so confident tells us what is out there. Feeling very comfortable with our abilities anymore? Yeah? Okay, now what's happening, right? We're hearing with our eyes is what's happening. Okay, why is this there? We live in a crowded world. Okay, we typically look at what's relevant. And our eyes help us interpret what's relevant. So in a crowded room, okay, in a party or a classroom, if you look at the person who's talking, you can kind of hear them better because you're using lip reading cues and everything. That's what's happening. This ability is there for a reason, but we didn't even know we had it, right? So what we think we hear, we are actually seeing, okay? All right. There's a cool Isaac Asimov story about this, where the word for, I don't know, stop and go is different by just one syllable or something, and it has planetary consequences. Okay, so now we're I hope not terribly confident about our abilities, okay, our sight, our sound, even our sense of touch, everything limited, right? Limited by resolution, by the range in which we can touch, the size, and yet we construct this full world out there. In my lab, in my research, one of the projects we study is the development of the circuitry, the nerve pathway that brings sensory information into our cerebral cortex. Okay, if our brain is in a bowl, which it is in our skull, how is it going to figure out the world? The nerves have to bring in the information. Nerves from the eyes have to connect to the right part of the brain at the back. Nerves from the ears have to connect to the right part of the brain. Nerves from our entire skin have to connect to the right part of the brain. And it can't be botched up, it can't be mixed up. Otherwise, we don't know what's out there. As it is, we are limited in our skills. Then if these nerve pathways got scrambled, we would really have no clue. And yet, amazingly, these nerve pathways make their way into the cerebral cortex absolutely with great precision and no difficulty, okay, when we are still in the womb. And then once we're born, with visual experience, sound experience, things get fine-tuned. This amazing feat is happening in every single one of us. Okay, it's happening in every little mouse on the street. Okay, cat, dog, mouse, monkey, whoever. All of them have to navigate the world. If a little species, if a little animal from a species did not figure out how to navigate the world, it's not going to be able to, let's say, gauge how far this branch is from the one I'm sitting on and when I'm jumping from one branch to the other. Okay, and unlike cartoons, we don't recover. In our modern world, we're not going to be able to cross the road properly. And any set of genetic defects that does not allow us to do these basic skills means that we don't get to be wandering around for very many days crossing roads, which means we don't get to eventually grow up and produce more babies like us. So any defects in these basic pathways, in these basic traits, get eliminated by evolution. Okay, if you can't see a tiger there and enough to kill it before it kills you, you're not going to make more babies like you who can't see tigers. It's harsh, it's cruel, but it's how the species survives. Okay? Now, having raised 2 kids myself, I often wonder how the human species has survived. I mean, nothing against kids, we were all kids, but my God, we are hopelessly unable to take care of ourselves when we're babies, right? How has the species survived? It survived with huge amounts of parental care. Okay, the amount of parental care human babies need. I mean, have you seen these lovely videos of baby horses? A horse just gives birth, the baby pops out, in 10 minutes it's standing on spindly legs and then it started running around. Yeah, a little chick pecks its way out of its egg, will lie there flopped and exhausted, and then will immediately dry off, fluff out, and then start going and pecking for food, for God's sake. Human baby? The survival strategy of the human baby is to look god-awfully cute. And it works. All of us have gone, yeah, I mean, parents will go without food, without sleep, all to keep this little infant alive. I mean, look at the baby's motor skills. Have you seen these really cute ones when, proud parents have taken it out for the first time, their first month or so, they're lying there in the pram, and every so often, it'll do. Have you seen that? All the muscles contract at once. Everything happens at once. Why? Why? Everyone do this, the good old okay. What contracted? Biceps. What had to extend? Every single muscular act requires something contracting, something extending, right? It requires coordination. Think about walking. Oh my God. Oh my God. Look at the sequential coordinated walking. Now, human babies, how long does it take them to figure out how to walk? And then when they start, this is my favorite. There's a reason why they're short, okay? It doesn't hurt as much when they keep. Okay, we all started out like this, okay? It's us. Now think of the horse. The minute it's born, it's up on 4 legs and it's actually running. Yeah. Okay, so this was about survival strategies that allow the brain to figure its world out and wire up. And for human babies, there's a lot of parental care involved. Okay, while the circuits wire up so that it can actually move, yeah, and so that its arms aren't unguided missiles that how many of you have been pelted in the eye by a baby hand? Okay, all right, that's the pathway I study. The sensory pathway that brings information about the world into the brain so that eventually the motor pathway can figure out how to navigate the world. Yeah, yes?
Yeah, since you derided the human cognition so much, if you think of a cognition index, is human species the best in cognition, or is there some other species?
Oh, he got me here. Oh, I'm only halfway through. He says, what about cognition? What about the cognition index? So I'm talking about sensory perception of the world and motor movement. We're not going into cognition. Okay, cognition is far, far out. In fact, it's so far out, it's out of this theater and out of my talk. Maybe we can talk about it afterwards. I mean, yeah, there's a reason our brains are so big. They do something beyond sensory perception and motor. But all of it is triggered by how we take in the world. Okay? Every cognitive activity we do starts with thinking about what is out there, right? And if the pathway that controls how we take in information is messed up, none of the rest of it is going to happen. So that's the pathway that we study. So let me show you one data slide from my lab, okay. I mean, this is work of my PhD student, Suranjana Pal, who just defended her PhD, so she's now Dr. Suranjana Pal. A delightful piece of work, and I'm going to show you one little jhalak, okay. So this is the entire nerves in the human brain, and they're color coded, you know, so you can see the directions and so on. Now, since nobody is giving me their brain to study, nobody? Fine. Sorry. This is a mouse brain. This is actually an mouse embryo brain. This blue is the cerebral cortex, and this green pathway is the sensory pathway that's carrying information about vision and hearing and touch and so on into the cerebral cortex, okay. And this is the pathway we study. Now, we study what genes are involved in getting this pathway to wire up correctly. What genetic mechanisms, because genetic mechanisms are what the embryo is born with, right. Think about an embryo in a womb, sort of like let's say mission Mangal that we have sent off to Mars or some planet. We've sent off this, you know, rover or something. Its job is to get raw materials from its surroundings and somehow create a computer that can control all of its movements and navigation. That's what an embryo does. It unpacks information that it has within it, okay, eats a lot of raw materials and basically wires itself up. That's how magical this computer is. It's a computer that builds itself through instructions that it came with. I wish our regular computers would do that. You open the machine and it's built itself. Oh my goodness, okay, but that's a developing brain. A developing brain is a computer that's wiring itself up based on the environment around it, okay. And this is why we need to give our babies as wonderful an environment as we can, okay. We need to give them stimulation and attention and everything so that their little computers are wiring themselves up to navigate their world. Okay, how would we figure out, how would we be able to identify what genes are, you know, important for this pathway to grow? Now, I'm going to do some genetic talk. Is that okay? Oh, you have a question?
When you're saying computer, that computers cannot build themselves, are you counting that we have made some artificial intelligence that can
Oh my goodness.
code?
Can an artificial intelligence program write the same code? Actually, very likely, but we're talking about assembling the hardware. So if you had to build a computer, you would go out and buy a motherboard and some chips and a hard disk, RAM chips, whatnot, you know, a video card, right? Otherwise, how would you be able to see? So you would buy these components and you would slot them into their slots and then you would wire things up, right?
Don't we do something like this?
We're talking about building the hardware. Maybe we'll talk later. Yeah. So we're just saying that you can't go out into a shop, the embryo can't go out into a shop and buy, oh, I'll get the better video display card. Okay. It's going to have to build a cable that carries information at the right rate. And in fact, the eye actually has a nice strategy. There is one pathway that handles color. Okay, that's a narrow bore pathway, signals don't go as fast, but they carry color and detail. And there is a wide bore pathway that travels fast, that's black and white. Okay, because if a tiger is coming at you or somebody's thrown a stone or a cricket ball, more relevant, okay, you don't want to know color, texture, all this stuff. You want to know where it is. Yeah, kaam ki baat, you want to know where it is. So the brain has these strategies. So it builds neurons that are big, cables that are thick, okay, and neurons that are small, cables that are small, that handle different things. So this kind of hardware has to be grown and wired up and controlled by genes. So how do we gauge how do we assess whether a gene is important, okay? Now forget genes. Let me say that you're a species that has never seen an alarm clock before. Actually, many of the younger generation may never have seen an alarm clock before. They are that species. Yeah. You know, this old style thing that wakes up the dead. Okay, suppose one such thing landed from the sky and you said, okay, this thing has a particular hand, the hour hand that's moving slowly, and another hand that's moving fast, minute hand, and a second hand that's moving really fast. And now you open it and you see all of these gears. and you say, I'm going to knock out a gear. Much like in the mouse we knock out a gene. I'm going to knock out a gear. And then we look at the clock and we say the hour hand is moving fine, but the minute hand is stopped. So we say, maybe that gear had something to do with the movement of the minute hand. Now, as scientists, we have to be very careful when we interpret this data, because the second hand may also have stopped, and we would not know whether the second hand is also dependent on that same gear or it's merely dependent on the minute hand. Yeah, so we actually have to apply this kind of logic when we interpret our data. What is our gene directly doing? What is it doing indirectly, and so on? But essentially, when we knock out a gene, this is what we do, okay, and this is how we interpret data. So here is the sensory nerve pathway of the normal mouse brain, okay. This is a low mag section, this is a high mag section, and what I'm focusing on is that green pathway I showed you before. This is an older brain now. The earlier pathway I showed you was in the embryo, just one thick fat cable. But now look, the cable has broken up and is now in these little dots, okay. And the high mag, you can see these dots, you can see each of these is a fiber. What are these dots? They're actually the connections where signals come from each whisker of the mouse. The mouse's whiskers are like our fingers. We explore the world with our fingers, right? So each whisker gets a dot. Because think about it, if you didn't allow 5 fingers to explore the world separately, if you put them into mittens, you're not going to be able to get your sensory resolution, right? It's like you have one fat finger. You need to keep the signals from the 5 fingers apart. Similarly, each mouse's whisker actually gets one spot in the brain. Okay, and this becomes a lovely model system for us to test, hey, what happens if you remove a whisker, and so on. This innovation happens after the mouse is born. You don't see these nice separate spots until the mouse is about 5 days old. Okay, now we want to test how might this pathway know how to grow normally, what genes might control it. So we have selected a particular gene which we expected would have a role in this process, and when we look at the knockout mouse brain, ta-da! Okay, it's very clear that the nerve pathway has reached the cortex, but it's unable to grow in. Very, very few fibers are growing in. So now this allows us to hypothesize and ask the next question, why is it not growing in? Okay, is this territory supposed to be receptive to nerve growth, and has it now become hostile because we've knocked out this gene? That's how we figure out what the gene does. So see, you're all scientists, you're all basically following the logic exactly, and this is what we ask our students to do. This is how we sit and discuss. Hey, what experiment would we do to test this hypothesis? Has the cortex become hostile? Has this gene now rendered it hostile? What is the gene doing to make it hostile? And so on. Okay. So this was a little glimpse of our science. We study how do you say it? We study the cabling system of the entertainment industry of our brain. Okay? Without this, we're blind, we're deaf, we can't see. Yeah? The only thing we'll be able to do is smell, because smell goes through another pathway. It's evolutionarily ancient, it goes through a different pathway. Okay? All right. Our basic circuits now are the same, right? We can all see things and move, but there are individual differences, right? Now, all of us can walk. Everybody got here, so presumably we're all able to execute the motor functions. But when some people walk, they look like athletes. When some people walk, they look like dancers. They're just graceful. The fine-tuning of motor coordination determines grace. Right? These fine-tunings are different in all of us. And these individual differences arise for 2 broad reasons. Some of these differences arise because they are just innate. Your genetic mechanism played itself so that the wiring just worked out with a slight difference. Okay, in some people, say the musical circuits hum more efficiently, right? And all of these circuits are affected by training and experience. That's called environment. When we say environment, we mean the experience of the environment. So if you listen to a lot of music, the first time you listen to a piece of music, you don't listen to it the same way as you would the 100th time. It's because you're paying attention to it differently, you're focusing on different parts of it. Okay? The same thing applies to anything. The first time you write a piece, the words you structure, anything improves with experience and training, right? So you're born with a certain baseline set of abilities, and within that you can change dramatically by either focusing on training or not focusing on training. And let me give you some examples of this. Let me first illustrate some intrinsic differences in all of us. How long do I have?
It's been 50 minutes.
50 minutes? Okay, we need another 10. Yeah, we need another 10 minutes. 10 minutes, everybody? Okay, innate differences. You've heard this before, I'm sure, or if you haven't, enjoy the ride. I'm going to play a soundtrack. The world is divided. The world is divided between the Yannies and the Laurels. Okay, and here we go. Sound, sound, sound.
Yeri, 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 Yeri.
Okay, how many heard Yanni? And how many heard Laurel? Yanni again? Okay, people come to blows over this. Don't. There's always, how could you hear this? I mean, come on, it's so clearly.
Can we go again?
Can we play it one more time? Okay. Sorry.
Yeri, 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 Yeri.
How many Yannis? Do you dare? You're in the minority. You can beat up. Okay, why do we hear this? Okay, why do we hear this? One of the best examples comes from this gentleman whom I've credited below. He simply changed the sound equalizer settings on this same track. Okay, and now I'm going to play it to you with techno settings.
Yeri, 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 Yeri.
Okay, how many were Yannis before and changed to Laurel? Okay, some people switched, right? How many were Laurels before and changed to Yanni? One sole person. Okay, let's do this one more time. Here now we're going bass, okay?
Laurel, 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 Laurel.
Okay, how many switched from anything you heard before? Okay, how many switched from Yanni either first two times to Laurel? How many switched from Laurel any of the first two times to Yanni? A different two. Okay, what is this telling us? This is telling us our individual perceptual circuits are different. In fact, some of us are so weird that when I did the test, I swear it was Yanni the whole time, and now I'm hearing Laurel the whole time.
When it was said that the other part is Laurel, I just tried to focus and find out, can I just hear Laurel? Still I couldn't.
You know, so the reason I might have switched between laptop and sound system is that the sound equalizer settings are different, right? Different frequencies get emphasized and not. You can Google this. There is a lot of explanation on this. Okay. My son introduced this to me. Yeah, that same son who was a little baby who was utterly. They bring interesting things to our lives. And my God, after hearing Yanni the first time, I was on Laurel for a month, and the kinds of conversations that would go on in our house would be, I've lost my Yanni, where's my Yanni? And then it's like, my Yanni came back. Yes?
Except from Yanni and Laurel, there's a third word, like Yorel.
Okay, how many unique individuals heard something different?
I heard a mixed Yanni Laurel.
We are all special. So when teachers in school say each of you is an individual, each of you are special in your own way, I have no explanation for this. I mean, obviously there's a perceptual gradient, right? And our ears try and fit it to whatever we think. Yes?
Is it because of the environment why we are having a difference?
Very good point. Is this genetic or environmental? Ouch. Let's take that in question session. I'm almost at the end.
Sorry, one question here.
Yes?
If you had not given us both the words, Yani and Laurel, and had just said Yani and asked us to hear,
Yeah.
is it likely that everybody would have heard Yani then?
They may have heard, so Yani and Laurel don't mean anything, right, to us. So they would have heard something which would be, the people usually hear the ya or the la. So since Yani is not even, I mean, Laurel at least we've heard the name, but Yani we haven't even heard. So people who I've done this experiment with without saying anything go, yeah, yeah. You know, they hear some year thing. So some of it is primed, yes. It was to illustrate that at any given time our individual perceptions are different. And since I changed the equalizer settings, obviously that's an environmental component, but your baseline settings are different in the first place, right? So let's just wrap up quickly here. This is training, okay? Nobody is innately born with skills to make chapatis or play tabla, right? Or ride a bike of any kind. But you get this with training, and that is motor coordination. Motor coordination is an easy way to demonstrate training, but it really applies to all our circuits, right? It applies to, I don't know, to composing things, to writing properly, to figuring out how to express yourself through any medium. Doing any element of your job properly requires training. And here we just illustrate it with motor, the motor component. What happens when you train? How many of you have tried any motor skills? Swimming, bicycling, chapati making, drums, whatever. My version is Kathak, okay, complicated footwork. And I find when I'm given a bit of complicated footwork, piano say, the day I learn it, I can't execute it properly, but when I sleep, something happens and I'm able to do the very, very fine changes the next day, okay? What happens is, suppose, you know, I have my weight on one foot and then I have to do the weight on the other, but then I have to keep the weight on one for a while. That mental weight shifting, that sequence clicks in better, okay? The mental weight, whether you're doing it with your fingers and so on, what comes after what? That sleep consolidates learning, that's a whole different talk, okay? So yes, sleeping before your exams actually helps. Wonderful tests have been done where students are given little words to memorize and then tested 8 hours later with sleep in the middle or no sleep in the middle. The sleep actually consolidates the learning, okay? It's a critical part of life experience. There's a reason babies sleep 20 hours a day, okay? There's a reason. They actually need that time to consolidate things. So motor learning can greatly change your abilities, which have innate differences to start with. And what's happening? Okay, what's happening? This is my last slide. How do we learn things? How does our circuitry enable learning and change? It enables it by actually changing the hardware. Existing connections can get strengthened if they are part of the new learning. They can get weakened or eliminated if they are not required as part of the new pattern. And new sprouts can grow. And this is happening in your brains all the time. Hopefully right now in your brains there's a whole bunch of new sprouts growing, or if you're dead bored, everything is like, okay. So let me show you a visual of a neuron growing in a culture dish. It's the most evocative description of how our brains are alive and plastic and growing, okay. So this neuron is going to send out many fibers. A neuron has input wires called dendrites that take in information, and then it has one output wire that goes to a target to connect, okay. This input dendrite phase I liken to the neuron doing Bharatnatyam. See? And then the one wire that decides its target is over there. Its target's over there, and it's going to grow and grow and grow and grow and reach the target. That's Kathak. So this is happening in our brains when our neurons grow. Okay? This is a real neuron growing in a dish. Okay? Sorry?
Initially it was taking in and then...
No. Initially it was simply sending different neurites, fibers, everywhere to sort of sample where it would make its connections. Okay, and these are the input wires, and then one of them decides it's going to be the axon, the output wire that's going to grow to its target. How it knows which side to grow. Okay, in the retina, how all the nerves know to find the point where the optic nerve is going to head out. These are questions that research students have answered over the years. How does a nerve pathway know which way to grow? How does a nerve pathway know to grow here and not there, but the other pathway has to grow there but not here? So these are questions that we actually answer in a lab. Okay, yes? What's the time scale on this? Usually a few days, 24 hours, 48 hours, by about 3 days you would see long fibers growing out. Depends on which neuron you start with.
But axon is the part that is growing.
Sorry? The long single output wire is called the axon. It's going to make connections to another neuron or to muscles. T-break, okay, well, these are the pathways of the brain, okay. I'd like to end with a little metaphor. This looks crazy, right? It's so many wires going every direction. It looks like Bombay traffic. But no matter how crazy Bombay traffic is, every car knows where it's going. Every car knows where it's going, and much like Bombay traffic, the nerves of the brain, when they are growing, all of them growing at once, they have long range signals, GPS. Continue on the sea link for 2 kilometers. Yeah, there's a long range signal. Then as they reach their area where they need to go, you turn off the GPS because there is local guideposts. Ah yes, turn left at this, car dealership, or turn right at this petrol pump. You'll recognize local guideposts. That's what nerves do. And then you reach, say, the gated community where there's a chowkidar, there's a gatekeeper. Okay, our gene, the sensory pathway gene, we think of it as a gene that controls the gatekeeper mechanism of the cortex. Without gatekeeper, pathway can't go in. So much like traffic, we have long range signals, recognized guideposts along the way, gatekeepers that allow this crazy traffic to actually make precise connections so that we can watch the movie that is our world and live our lives in it. Thank you very much.
I know that you have lots of questions for Shubha, so please come back soon.
The brain needs glucose and nutrition.
till 5 o'clock we will have lots of things. Usually what we do, we have things happening in the morning, repeat in the afternoon, so you can come for a half day, whichever is more convenient, especially for lab visits or something. You can maybe do lab visits in the morning and go to the Mela outside in the afternoon or vice versa, whatever you like. The lab visits require you to sign up. We will start the sign up thing once we know which labs are on. The rest of the stuff in the which is open air, you can just come anytime and walk in, no problem. That's happening on the 23rd of February.
But kids below 8 will not be allowed inside the labs.
Yeah. Labs we cannot take, very teeny bits. You'll grow up and then you'll come to the lab. Don't worry. Please come and enjoy the Mela outside. So you should be at least, I don't know what, at least this high or something to go to the lab. So there is something we put up. I think it's age 8, 10 or something. I've forgotten what it is. Don't worry. Yes, there is a session on the next session is Ruparel. Today is 5. So no, that's not the third Sunday.
That's fourth Sunday.
That's the fourth Sunday.
Okay.
So it doesn't, Chai and Wai happens as usual. This is the National Science Day program at TIFR. Just a reminder, 2 weeks from now, the session at Ruparel College is it 2020 yet? And that's about calendars. Okay, it's about calendars and counting and things like this. Done by a mathematician, Sagar. He's done lots of programs on origami and other stuff before. Also works on calendars. And if you are not on our mailing list and you'd like to join us, well, 2 things. You can just follow us on Facebook or Twitter at Chai and Wai, or send an email to outreach@tifr.res.in. We'll add you to the list. Okay. I'm going to let Cyrus take over the question answer moderation because my throat is on its way out. So, yeah. If you have a question, raise your hand. We'll try and get as many as we can. We'll have to answer them.
In moderation. Moderation. Can't buzz any sense too much.
Okay, here's one. We can start right here.
Okay, we take 1, 2, 3.
So how does all this connect to yoga and meditation?
How does all this connect to yoga and meditation? Okay. The very short answer, I don't exactly know, and I'm not sure anybody does, but I will give you my best educated guess. As you can see, we have a lot of circuits in the brain. Okay. My best guess is that our circuitry operates better if we were able to focus it, streamline it, not have too many random distracting things happen. My best guess is that meditation or yoga or focusing on your breath allows you to calm errant circuits and streamlines the functioning of what you have. And with practice, you can get better and better at this. The evidence is clear. People who meditate, people who do yoga have a level of performance that is better than before. So my best guess is that it simply makes your circuits function in a more streamlined, less distracted fashion. So we haven't been able to explain it yet, but I think we will, and that would be one of the goals of neuroscience.
Just a sec. We take a quick one question per person, so please keep your question prompt. We'll take one because he's been holding his hand up quite tirelessly. And Vishnu, go ahead.
When I watched that video, I noticed that sometimes, what the main cord, sometimes some fibers are branching out of it, from the main cord itself. The answer cord.
When the neuron was growing, the fiber that was growing this way, it wasn't just heading there, it was going tweet, tweet.
It had
many little fibers coming out. Oh, from the main neuron itself?
And then they moved forward.
It had little boluses going through the cord, through the fiber, right? There was little. It was moving in a certain direction, but along its length there were little side branches. Axons can send out side branches. It was very sharp-eyed of you to catch that, called collaterals. Sometimes a neuron will connect to 2 things. In fact, when a neuron connects to a muscle, at the very end, it connects to many, many, many things. And the better a neuron connects like this, the better it can fire that muscle. So, sending out side branches is a natural function of sending out an axon. Now, this was a culture dish. There were no other neurons around to connect to. But this particular target was, this particular neuron was heading towards one target. So it was an artificial situation. Those side branches didn't get very far. Okay. Sometimes when a neuron is growing in one direction and a more attractive cue comes somewhere else, either that main fiber will turn or a side branch that was little before will become stronger and stronger and this one will retract. All of this happens in the brain. All of these are natural mechanisms and it was very, very sharp-eyed of you to pick it up.
So I had made an observation on one of the slides that you showed. It was the one that the gentleman says ba or fa. And I could be completely wrong, it could be just my perception. But if you could just play it again.
We don't have time to play the ba fa again, but just tell me what you mean.
Notice where you showed both the images.
Yes.
One of the gentlemen was, his lip movements were quite different from the other. So I guess the upper lip, his teeth could be showing in one
so
and the other it was both the lips closed. So what I'm trying to say is that I think there could be different words that he was speaking.
So actually that is exactly the point. Yeah. The Mcgurk effect, which you're asking about, is that the Mcgurk effect is when the lip movements override what the sound is. So he was going with one set of in one video and with the other video. The soundtrack was actually ba 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 the whole time. But if the soundtrack ba 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 is matched with lip movements going, the whole point of the Mcgurk effect is that you would hear the first sound.
So it wasn't that he was saying that, it was just a soundtrack.
So that's why I invited you to close your eyes during the fa 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 video, the apparently fa fa video, because if you close your eyes there, you would hear ba ba, and when you open, you hear fa, no matter how many times you do it. You're just programmed that way, there's no escape. I've heard this video dozens of times and I can't escape it.
Thank you. Yes, you had one question back here.
So at any point of time in future, is it possible for neuroscience to replicate the exact circuitry of a brain? Is the knowledge absolute regarding what brain is at any point?
Okay. Is there a possibility in the future that neuroscience will be able to replicate the exact circuitry of the brain? This actually was a goal when people started out, but you know what? Artificial intelligence programs have already kind of done it. Okay, where's my AI friend? Nobody understands how machine learning actually works inside the black box that does it, much as nobody understood much of what the brain did some time ago. And yet one has to ask, what is the goal that motivates this kind of study, right? I'll give you a metaphor. If your goal is to fly through the sky, you might start looking at birds which have feathery wings and flap, and you might say, okay, let's do it that way. And you might tie feathery wings and try and flap and jump off cliffs and so on, and those efforts might meet disaster, right? Or then you might say, okay, let us achieve the same effect using completely different technology. Let us have jet engines to compress air and have this, flow over a wing that doesn't flap. And you would actually achieve liftoff and flight. To my mind, that metaphor is how the brain flies and how artificial intelligence programs fly. So if your goal is to achieve higher function, you don't have to do it the way the brain did it. Evolution is not the most efficient way of creating anything. Okay, evolution cannot throw something away unless it prevents the propagation of that species broadly. So evolution adds on and adds on and adds on things. So it's like, oh, this is not working well. Well, you can't really throw it away, but let's add on this to kind of make it better. It's like the Hubble telescope where the image was fixed by putting in many, many more mirrors. That's sort of broadly evolution. It makes things very complex and very, very, very messy. Eventually you get function, but it may not be the most optimal way.
Gentleman over there in the back.
Yeah, we have seen people with extraordinary or maybe striking capabilities, mentalists, people who can look at a distance and tell you exactly how far things are or guess the weight of a person maybe to the last key.
My mother would look at that and say that's maybe 4.5 feet, and man, she was right. Yeah. She could fit in boxes into spaces, I mean, just.
So considering these conditioned or learned states of categories or types you find in the circuitry. Is there a kind of broad typologies that have been established?
So let me just try and rephrase to see if I understand it. We see people who are much better at a particular skill or skills. How many different such categories are there, and is there a brain circuitry correlate?
In the circuitry type,
Okay, it's impossible to, we're at the stage of asking how a mouse can run a maze. Okay, how a human can estimate distance and so on. I mean, just to set up that kind of experiment, and then you would have to eventually dig into the human's brain. So it would be near impossible to have a fine-tuned understanding of if this wire goes. What you want is if this wire connects to this neuron, then you're going to be able to measure distances properly. And if this wire connects to that neuron, maybe you'll have perfect pitch.
No, for example, a study of Einstein's brain, only the glial cells were low, that's all. Something of that kind of categorization, is it possible?
I think you're answering your own question, right? I mean, the poor man is dead and we still haven't figured out what his brain is. There are a million different Einstein-like capabilities, right? There are, I mean, every computer is tuned in its own unique way. And to sort of digress, take your question into something that's close to my heart. I think it's our job as adults, parents, to help our little brains figure out what it is that they're genuinely good at, because then they're going to perform at their best and be fulfilled, and so the possibilities are endless. A friend of mine whose child was put down in school, not attentive, not this, not that, and she tried to approach the teachers and say, look, my child has a reading disability and so on, and we need to help him this way and that way. And the very unthinking teacher's response was, your child is not Einstein, how far is he going to go? I mean, imagine, right? And her response, which is something that struck me, is, my child is not Einstein, my child is, I will just, my child is whatever, Salil Karwal, and you don't know what he's going to become. Okay, so that really summarizes it. You really don't know how each individual brain is going to flourish, and we should nurture that.
Gentlemen, the last one.
You mentioned the limitation of the five senses. What is the neuroscience take on this thing called the sixth sense?
That which
some evidence to suggest how you can augment the five senses too.
That which we don't understand is called the sixth sense. So the question, there are five senses, what is the sixth sense? So that which we don't understand. Look, there was a time when people thought lightning was an act of God. Okay, now it's no longer an act of God for most of us. Have I offended anybody? We know it's electrons. So the brain is complex enough that much of its functioning we don't understand. Classically, mothers are thought to have eyes at the back of their heads, and there are many mothers who function like they do. Now, I mean, we know that's not a sixth sense, right? We know that it's a complex circuitry in there that's attuned to the behaviors of her child, and that she knows that the child is in 2 minutes going to get bored of this and jump into that, and looks at just the right time. Yeah, we mothers get our kicks like that.
Neuroscience actually done any research in this domain to explore? I mean, from that Western yoga, the Western.
So has neuroscience done any research into this? All of these questions are in the space of absolute fine-tuned, extraordinary detailed functioning of the brain. We're at the stage of asking, how do the nerve pathways cable so you can see? Okay, how do we discriminate between shades and colors? How do we fold? So we're asking questions at a much more basic level. These are sort of microfine individualities. I cannot tell you, nobody can, what makes you you. But certainly there's a whole lot of unique circuitry that does. So eventually we'll get there, but we're at a fundamental stage of asking the broad functionings of the pathways.
We have a gentleman over there. You can keep your answers short so we can get more because we seem like we have a lot of questions.
Okay.
What's hypnotism then? Hypnotism means all the magicians kind of make 100 people believe in or look the same thing. How do they control hundreds of people in a certain way?
How do magicians achieve hypnotism? Very short answer, I have no idea.
Okay, there's a question over here. The little girl had a question.
When a female horse produces a baby, it just runs, but why don't humans run when they are born? Are we lazy?
When a horse produces a foal, it gets up and runs right away, but human babies don't run. Are we lazy? The human babies are born when their muscles are not properly connected up to their nerves. The circuitry of the body is not hooked up properly. Because human babies have parents who are willing to do everything our parents did for us, right? Human babies are born when the brain has reached a size, so all of the effort goes into the brain when the human baby is born. The body is small still. When you draw cartoons of human babies, you draw big head and small body. So the baby is actually pushed out when the brain is as big as it can possibly get, and yet the baby can be born without killing the mom. That's the limitation. If the baby got any bigger, then it would not be able to emerge. So a lot of brain development has to happen after the baby is born. And that's why babies lie around and do the best they can, looking god-awfully cute, so that parents are willing to clean all the poop and deal with all the, so that the little nervous system will grow into becoming a big nervous system.
I have a follow-on question to that. Does that increased plasticity contribute to better learning and development later on? Is there an advantage of having that stage of?
Is it advantageous to be born very, very immature so that you can learn more skills and so on? That's a very interesting way to look at it. And now I'm suddenly thinking positively about my teenager. So the teenage period is yet another period of a lot of flexibility. So much is going on there that nobody can figure it out. I suppose it is all eventually to the benefit. So that's a very nice angle. Thank you. That's why I love giving these talks. You learn from everybody.
Gentleman over there.
Is it true that with ESP you can bend objects?
Is it true that with ESP you can bend objects? So I would say that's like lightning is an act of God. Okay. Either science has not explained it yet and it will eventually, or the data is not actually real. There is some illusion happening there. Either one of those two is true. I don't know which.
About that Yanni and Laurel thing, or the effects related to that, does it have anything to do with why some people think that accents from other places are weird?
Yeah. Does Yanni and Laurel allow us to explain why people can't understand people with accents who come from different places? Absolutely. Auditory perception, to hear words, you don't actually hear the way a microphone might hear the entire spectrum, you categorize the frequencies you hear into syllables that you're familiar with. So it's like putting your sound through a sieve where there are only a few holes, and this is learned. So a baby will hear many, many different sounds, such as in India, we can all hear the difference between per and bh. But most people from only English-speaking countries will not hear per, bh. They will hear the non-aspirated version as p, and the aspirated version as p, and the non-aspirated version as b. So my name, Shubha, the best my American friends can achieve is Shubha, because the H sound is available only with the P for them. And they hear it that way. Okay, the same thing between the Japanese L and R. Okay, they cannot hear L and R differently after a certain age. So you group things into the perceptual sort of sound nuggets that you're trained with. And anything different is going to be a little odd, and then you force it through one of these.
Lady in red over there.
Could the circuits be rewired, meaning once it's set by genes?
Can what be rewired? Can circuits be rewired? To a good extent, yes. For example, so okay, if you are right-handed and you fracture your right hand, you'll be really bad with your left hand for a while, and then you will get better. And what's happening is a lot of rewiring is happening there. People who don't have hands, have you seen these amazing artists who paint with their feet? So the cortex that is dealing with hand movements gets taken over by the cortex that deals with leg movements, and it's like devoting more processing power. It's like if you want to take a high resolution picture, you'll buy a many more megapixel camera. If you devote more bits of cortex to something, you're going to be able to have more power to process it. So yes.
Gentleman in the black sweatshirt.
So you talked about difference in perception among different species. So is the perception of time also different? Are the days longer for an ant?
Wow. Are the days longer for an ant? Do species perceive time differently? It's a fabulous question. Now, seconds, minutes, and hours are a human construct. Species born on Earth have one clock, the sun. And with the day and the night, all the species on Earth are programmed in a circadian rhythm. Oddly, okay, this was a great, a Nobel Prize some years ago was given for discovering the circadian gene. So if you Google it, you'll find some very nice stuff. I'm sorry, I'll just get some water from a backpack that I had left somewhere around. So each species is programmed to the circadian clock, but it's not hardwired to it. It can be entrained. The human rhythm is, I think, thought to be 25 hours. But every day the sun comes up and programs us. So if you do experiments with humans, I think undergrads who are willing to do anything for free food, in a room with no external input or sensory experience, I think they will end up cycling on a clock that is longer than the regular one. So you can be programmed, you need to be entrained though. Likewise, species are all entrained with the sun. The neurons that do the entraining are amazing. It's called blind sight. There's a set of neurons in the retina which you do not use to see with because you're not actually processing visual signal from it. These are in the far blue region. And these neurons are the ones that that take in the circadian signals, and you need them to reset your circadian clock. So if in a mouse you eliminate these blue sensitive neurons, the mouse is no longer entrainable by a new circadian clock. Okay, and yet these neurons are not used to see. Now, how many of you are really troubled by this horrible blue LED light that shows up in at least my air conditioner? Okay, there's a reason that blue light troubles you. This sharp blue light and the blue thing from your gizmos, that messes up your circadian clock because it speaks to these very neurons. So put those yellow filters in, get rid of the blue, you'll be very happy.
Lady on the last one.
This is a follow-up of the Yanni question earlier. Is there a Yanni type and a Laurel type? Is there any implication to why some people hear this and not the other?
Your question is, does the Yanni-Laurel experiment help us understand why people hear one or the other?
Yeah, and are there any special abilities or capacities tied into one or the other? And it's
Okay.
amazing that can happen, right?
So,
We can particularly hear that.
so go home and play it again on your mobile phone and play it again on your laptop. And were you a Yanni or a Laurel? Okay, I promise you'll get your Laurel sometime. So a given circuit, I used it to illustrate that at any given time we're all different, right? But because this is a perceptual thing, not really a sensory thing, it's going to be changeable based on context.
Sure, I just wondered if there is something that predisposes people to listen to one versus the other.
Is there, okay, thank you, I understand better. Is there anything in your circuits that predisposes you to listen to one or the other? You know, I've done some reading on this. Do younger people hear more frequencies? Do older people hear less frequencies? I have not found a correlation in all of the times I've done the experiment. So this one may not be. It is a fact that as you age, you don't hear very high frequencies. But this experiment does not discriminate that. Yeah, yeah. Oh, yeah. We had some unique specimens right here. Yes, what did you hear?
I heard both, like as if two people were saying.
You heard two, you heard like people were saying Yanni and Laurel in your ear at the same time? Come to my lab tomorrow. No, so people do hear mixed sounds. People do hear mixed sounds, so.
Young scientists are like that.
Yes?
When we grow up, why do forget more when we are small age? When we grow up, why do we forget more? And when we are small, we don't forget.
Why do we forget when we grow old, and when we are young, why do we remember better? You know, my kids ask me this all the time. The brain works better when it's young. When you're born with most of the neurons that you're ever going to have. Can you imagine? In your head, the neurons that you have will be with you even when you're 70 years old. Most of them at any rate. Those poor tired things, they've been working so long. Their ability to hold on to connections and so on weakens. But you know what? Memories that you make when you're young will be fresh and with you forever, especially if you keep reminding yourself of them. So the more often you remember something, the better you'll remember it. And things that you remember when you're a child, you'll remember when you're grown up. So I look at my kid and think of all the junk he's filling his head with. The kinds of things he remembers, the micro details of things. Oh my God, every statistic of every cricket match ever played. And I'm thinking, save those neurons, you'll need them later. Yeah. But this is what he chooses to remember. I mean, you know, every book he's read, he remembers every single thing that anybody ever said to anybody in that book. It's incredible, and you're probably like that. All young minds are like that. So enjoy yourselves. You know, there's this lovely little song. You want to sing with me? Enjoy yourself. It's later than you think. Enjoy yourself while you're still in the pink. The years go by more quickly than you think. Enjoy yourself, enjoy yourself. It's later than you think.
Longevity in life that we are having.
I'm sorry?
Longevity in life.
Yes.
I mean, the lifespan is increasing. The biggest challenge which is expected to come up is on Alzheimer's. So how can neuroscience, I mean, whether it is helping or how is it helping?
Longevity and Alzheimer's. We are living longer, but our brains need to hang out with us so that they're able to function. How is research helping that? Alzheimer's research has gone very, very far. People have some idea of what happens in the Alzheimer's pathology. So basically, Alzheimer's is like when the hard disk gets corrupted. Okay, bits of the brain begin to. So broadly, two approaches. How can we detect it early so that we prevent the decay? And how can we help stimulate what's left? So those are the two approaches growing. And medical science is a lot better now than it used to be. So early diagnosis is actually the best way to do it. So early biomarkers of Alzheimer's are being pursued. Some have been identified. How early can you detect it seems to be the key. There are some genetic variants. There are some non-genetic variants. So we're working on it, hopefully before I age.
Correcting the corruption which has already happened?
For correcting the dead neurons, unfortunately, once a neuron is dead, it's dead. Unfortunately. But other neurons can take over. So if you catch it early, you catch it before too many have died, and then you can get your skills back. So people who've had, say, stroke, literally small bits of the brain die. For a while, you know, one side doesn't work and so on, but with practice and exercise, it actually comes back. What's happening? The remaining neurons are growing more fibers.
There is a small addition to this book written by Dr. Terry Wall. He's a neuroscientist. She has written a book on Mind Your Mitochondria. If you keep your mitochondria safe and you can still develop them, the diseases will not come. In fact, she herself was afflicted by MS and she got totally cured and now she's cycling. So it's possible. It's not impossible. What you said is that the brain cell keep on developing if you keep the right diet.
Thank you. Thank you. The gentleman alluded to the mitochondria as the site for many therapies and when there are problems with diseases. That's indeed true. Thank you. The mitochondria, the energy powerhouse of the cell, is sort of linked to your metabolism, how much nutrients you have, how much damaging chemicals you have, and so on. And the mitochondria is actually a sensor, and if that starts going bad, many pathologies develop. So the powerhouse of the cell, exactly.
Lady in the third.
You said we perceive the world through the five senses we have. What is your take on the phantom limbs and phantom pain which people experience? Are we trying to force ourselves into an imagined reality and making our existence itself a pain?
Oh, very nice. The question is about phantom limbs. Are we imagining things? So phantom limb is when, if you have an amputation, the person still thinks the arm is there, so it's a phantom limb, and indeed they will feel itching in it or pain in it and so on. Here's my best understanding of this. There's a lovely book written by V. Ramachandran, Phantoms in the Brain. So our best understanding of it is essentially the nerves that give you sensation from the amputated limb, okay, they, those nerves connect via relay stations to the brain, where the neurons that sense the limb, they are still fine. They just haven't figured out that the sensation isn't actually coming from here anymore. Okay, so anything that stimulates those neurons is now going to give you a feeling that you have sensed your little finger, which isn't there anymore. Now, if this piece of cortex is no longer getting signals from the amputated limb, the neighboring, nerves that are, say, normally take signals from other places are going to take over that bit of cortex. Okay, so in fact, V. Ramachandran found, basically a map of the amputated hand on the cheek, where turns out nerves from the cheek were connecting to the cheek part of the cortex, but also the amputated hand part. Yeah, so touching this would give that signal. So the phantom is created by the fact that nerves from different parts of the brain take over the cortex that is now spared from the amputated limb and give you the sense that that limb is there. That's sort of one little aspect of the whole thing, but I mean, read the book. It's,
Why is it that the brain is forcing itself into that kind of a mode?
why is the brain forcing? So the brain doesn't know what it's doing. Nerves grow where there is space. So if a nerve takes over an area which once gave you the sensation of your finger, then that area is going to say, hey, finger, finger, finger. Sorry, that's a really bad, sorry. Wrist, wrist, wrist.
We have our last three questions. I've already mapped them out, don't worry. The lady in the last row.
Tyrant.
So this is a follow-up on CAN circuits with Rewire. How important a role do the chemical guideposts play in the switch of this rewiring of the circuit? And imagine if the circuit follows path A. If you switch to path B, won't there be any interference from the chemical guidepost of the original pathway? And so how does that?
Okay, the question is about rewiring circuits and the role of guideposts. Now, guideposts play a role typically in development, when in the embryo the whole thing is being laid out to start with. When you talk of rewiring, the circuit has already gone past the guidepost, and now you're talking about a mature neuron whose axon has reached here to now send a collateral and go there. Okay, so those processes do not use the guideposts and so on that were part of the embryonic pathway. The embryo is a small place. So luckily the major mapping is done in a small place, and then it lengthens, and there's a whole area of science in asking, You know, how do you lengthen an already laid out circuit? You know, when a baby giraffe is growing into big giraffe, how do you put in the new wire? There's a whole area of science that looks at that. And rewiring does not directly use those embryonic mechanisms.
So, how do the mechanisms underlying various neurodegenerative diseases differ?
How do the mechanisms underlying various neurodegenerative diseases differ? I'll give you a short answer to schematize it out. It simply differs on which cell goes bad or what in the cell goes bad. Which cell goes bad? For example, disorders of movement, you could have the muscle cells going bad. If the muscles themselves atrophy, you're not going to move. Or you could have the nerve cell that's talking to the muscle cell go bad, so it's not sending the signal. Or you could have the insulating sheath that's wrapping the nerve go bad, so the signal is not true and pure. So depending on which cell type you attack, okay, so it can be that broad, and then of course the mechanisms that make the cell go bad in the first place.
And we will have over there.
When we did the hole in the hand exercise, after that you asked whether how many of us thought it was real, how many of us thought it was imaginary. So I wanted to ask, how do you define reality, especially after you told us how our perceptions are formed? How do you define in that context?
The hole in the hand, real versus imaginary. The question is, after this whole talk, how do you define reality? Thank you. Thank you. That was actually the point. Thank you very much for it. The point is, everything we see is as real or as imaginary as the hole in the hand. Everything is brought in through the same senses. Everything, if you wish, is a figment of our imagination. The old philosophers, the whole world is Maya. They were bang on. It is. I mean, what is out there? It depends on who's looking and what you're looking with and what you're using to process and create this construct. So absolutely, you know, so if at the end of the day you're completely destabilized as to what you really know about the world.
Well, so
Okay,
one unfortunate.
actually, the reality, I don't know about her reality, but I know one reality that it's 1 o'clock.
Is it?
Yes. on Prithvi's clock it is 1 o'clock. They have other shows here and they would need to use a space and they're kind enough to let us use it on Sunday mornings. So unfortunately we must end this. I know you could go on and on and on, but thank you so much for Thank you all for coming. You can always send us questions by email or something. If we find them interesting enough, we will get back to you.
And thank you to all the young minds. Keep those circuits growing.